Hi, I've worked in preclinical drug development for over 30 years, and I'm often asked by European, Asian, and North American sponsors about the US FDA's 505(b)(2) process. This presentation will aim to answer many of the more regular questions that I get asked, and in particular to address implications for the non-clinical program. I'm going to start with some of the basics, then I'll talk about when a 505(b)(2) route can be used and when it can't. I'll explain some of the significant advantages and how a simplified preclinical package can be used to support the submission for a range of different situations, some of which are less obvious than others. Lastly, I'll touch on some of the more important things to consider in terms of the wider business strategy and some of the early interactions with the FDA. So what is a 505(b)(2) submission? It's a bit of a mouthful. Well, it's a US FDA regulatory pathway, which, unlike a 505(b)(1) submission, which is used for drugs with a novel API or active pharmaceutical ingredient, it relies on safety and efficacy data from a previously marketed product. Importantly for sponsors, what this means is that some of the key studies do not have to be conducted with a new product by the sponsor. They can rely on existing data for the marketed product. So let's consider when you can and when you can't use the 505(b)(2) route to see if it might be right for you and your project. The route cannot be used for completely novel drugs where the API has not been included in a product marketed previously in the USA. But you can use it when a drug using a previously marketed API is used for another indication. You need to bear in mind in these cases that some typical studies may not have been included in the original package. For example, genotox may not have been included if an API is previously approved for an oncology indication. And these studies might need to be conducted for the new indication. A 505(b)(2) approach is most commonly used when there's simply a change in the formulation, dose strength, or the route of administration of a drug. However, the approach can also be used for new prodrugs of existing active ingredients and to provide some of the required data for combination products where one or more of the APIs have been used in a marketed product. So we know when a 505(b)(2) route might be used, but why would you want to use it? Well, there are significant benefits to be had by using a 505(b)(2) route with four main reasons. Because you can rely on pre-existing safety and efficacy data, fewer preclinical and clinical studies are required, which means that development costs and timelines are very significantly reduced. Also, and critically, because the API has already undergone extensive testing for both safety and efficacy, these programs have significantly lower risk than the development of a novel drug, so failure rates are much lower. Despite these advantages, many products also qualify for significant market exclusivity periods, all of which helps to explain why in 2024, over 40% of all new FDA approvals took this regulatory route. When you compare the 505(b)(2) route to the 505(b)(1) route, which is that's used for drugs with a novel API, and with the ANDA route, there are significant differences in terms of exclusivity, studies required, and costs. As you can see, the 505(b)(2) route does allow for market exclusivity in certain circumstances, such as for orphan indications or where it will be used in children. So if this is the case for your project, it could be very attractive. Cutting to the chase though, the bottom line of the table shows significant cost savings. Please also note that the figures on the table are very much on the conservative side. They include orphan drug development programs where costs can be significantly lower than those for non-orphan indications. So in many cases, the cost differences can be significantly higher than indicated here when looking at indications with larger patient populations. So the savings might actually be significantly greater. So how will taking a 505(b)(2) route impact what is required for a preclinical safety program? Here are a few common examples for illustration. In general terms, some pharmacology and PK studies are still likely to be required, but the safety package will be much less extensive than that required for a novel drug, and will likely not include safety pharmacology, genotox, or reprotox studies, to name a few. Repeat dose tox studies may also be of shorter duration, and in some cases may only require histopathological assessment of locally exposed tissues rather than the full standard tissue list. This can significantly impact the cost of the study. However, you should consider carefully what excipients will be used in the formulation, as novel excipients may require further safety testing. That said, preclinical safety packages can typically be significantly reduced in scale. Let's look at an example where a new formulation has been developed for a dermal application. In such cases, there may be a risk of delayed hypersensitivity, possibly ocular irritation or phototoxicity. So studies, often in vitro, will be required to assess these risks. To assess dermal toxicity in vivo, a tox program with dosing up to 9 months may be required, but often only in a single species, and only where a drug is going to be used chronically. So as the best model for human skin is usually mini pig, that's likely to be the species of choice for the general tox studies for a dermal program under the 505(b)(2) route, assuming that metabolism in this species reflects that in human. For a product where the route is changed to IV, chemocompatibility studies will be required. However, if systemic exposure is the same or lower than the marketed product delivered by another route, it's unlikely that further in vivo toxicity studies will be required, making for a much smaller preclinical safety package. Like the change to a dermal route, the preclinical study is required to support development of an ocular formulation where systemic exposure will be the same or less than a marketed product for a different route, would focus on the tissues at the site of application where exposure is likely to be higher. The ocular toxicity studies will assess systemic exposure using TK assessments. This is to confirm there's no greater exposure than the marketed product. And an assessment of ocular distribution will be required to confirm which tissues are likely to be exposed in clinical use. If no greater systemic exposure is confirmed from the TK data, then assessment of locally exposed tissues only from the toxicity studies may be sufficient. For intranasal products, 2 to 4 week toxicity studies in 2 species will be required to get into first-in-human clinical trials. Chronic tox only needs to be conducted in the single most appropriate species, with a duration based on the proposed clinical use. You should be aware that where a product has a particle size of less than 5 microns, the inhalation tox may be required due to deeper penetration into the lower respiratory tract. In some cases where intranasal exposure data is available on an inhaled product, the kind of originator product if you like, intranasal tox studies may not be required, although you should be aware that often this data is not available. I've worked with a few companies developing extended release products, often to improve patient compliance. In these cases, toxicity studies in 1 appropriate species is usually sufficient. But be careful, it should be the case that the duration of the toxicity study covers the full duration of the enhanced release profile. Where excipients are novel for the route of administration, their biodistribution also needs to be assessed. So I've provided a few examples of where a 505(b)(2) route can be used and how the preclinical package can be streamlined. Let's look now at how you might evaluate if the 505(b)(2) route is suitable for your project and what the major steps with the FDA are. Picking the right candidate is key. There needs to be a robust scientific assessment to confirm that you have a good and scalable, homogeneous formulation which is not prohibitively expensive. The product needs to fulfill a defined medical need, and the product, including excipients, needs to be safe. The sponsor should consider what studies will be required and what labeling requirements and exclusivity are likely to apply before they go ahead with the project, and certainly before approaching the FDA. Lastly, a commercial assessment should consider the potential market, whether competitors are developing similar products, and what price might be achievable. Once the decision to go ahead has been made, a pre-IND meeting is typically the first contact with the FDA, and the primary purpose of that meeting is to get FDA's approval of the chemistry, manufacturer and control, or CMC strategy, and the clinical plans. The meeting is usually held when the sponsor has early formulation and stability information and some key non-clinical data. The proposed clinical protocol should be included with the briefing book alongside a comprehensive CMC package. Once FDA agrees the plan outlined in the pre-IND meeting and the required preclinical studies are completed, the IND can be submitted. So, in summary, 505(b)(2) represents a quick, cheap, and low-risk route to market. The approach can be used for a range of products, but only where the API is already marketed in the US, and that data can be referred to. It's most commonly used for a change in dose route or formulation, but the regulatory route can also be used for new indications, for combination products, and for prodrugs. Altasciences design and run a lot of preclinical and clinical studies for 505(b)(2) submissions, and we'd be happy to talk to you about your future program. Thanks for listening today. And if you'd like to contact Altasciences to discuss your project, you'll find the website address and contact details on this slide.